Hello and welcome to Spotlight on Antibodies, a podcast from Biointron, where we bring you the latest antibody industry trends. I'm your host, Jovina Celestio, and this month in January, top news includes glycoengineering, anti-malarial antibodies, and PROTACs. According to Research and Markets, the global market for FC and glycoengineered antibodies is projected to be worth $38.8 billion USD in 2024. Over the next decade, the growth in opportunities for drug developers is expected to be driven by an increasing patient population and rising demand for these advanced antibody therapies. Glycoengineering, the process of modifying glycan structures on proteins to enhance their functions, plays a crucial role in improving antibody efficacy. It is particularly significant for modifying the N-glycans on the Fc domain of immunoglobulin G, which influences key immune responses. This field has advanced significantly, with strategies such as mammalian cell expression system engineering, chemoenzymatic techniques, and yeast-based approaches enabling precise glycan remodeling. These methods improve the homogeneity of glycans, simplifying the study of structure-activity relationships and enhancing therapeutic efficacy while minimizing side effects. Additionally, glycoengineering has been pivotal in developing antibody-drug conjugates, or ADCs, offering new opportunities to optimize IgG-based therapies. On April 30th, 2024, China's NMPA, National Medical Products Administration, approved benmelstobart, or Andoway, a humanized anti-PD-L1 IgG1 monoclonal antibody that was Fc engineered to reduce effector function. Other glycoengineered antibodies that were previously improved include AstraZeneca's benralizumab, Genentech's obinutuzumab, Incyte's tafasitamab, and Amgen's inebilizumab. A recent review highlights the role of glycosylation in monoclonal antibodies for cancer therapy, emphasizing its impact on efficacy, safety, and pharmacokinetics. Combining genetic and metabolic glycoengineering has enabled precise tuning of glycoprofiles, improving antibody-dependent cell-mediated cytotoxicity, or ADCC, complement-dependent cytotoxicity, or CDC, and target specificity. Advances in combinatory glycoengineering, such as afucosylation and glycan modifications, have optimized therapeutic outcomes in clinical trials, including immune checkpoint therapies targeting PD-1 and PD-L1 glycosylation. Emerging strategies and high-tech tools for glycan detection and analysis promise to revolutionize biotherapeutics, enabling the production of safer, customized glycoproteins with enhanced efficacy. Various strategies for glycoengineering antibodies have been developed, including from a recent paper describing endo-SC D234M, a mutant endo-beta-N-acetylglucose amidase, as a platform for enhancing the therapeutic efficacy of monoclonal antibodies through improved transglycosylation activity. It remodels diverse glycans on monoclonal antibodies, producing homogeneous antibodies with 326-fold increased ADCC activity. High-resolution crystal structures of endo-SC D234M, including its complex with the G2S2 oxazoline intermediate, reveal the oxahole, a critical structure feature stabilizing intermediates and catalyzing transglycosylation. Key interactions, such as hydrogen bonding in the oxahole and contributions from loop structures, elucidate the mechanism of substrate selectivity and activity. Mutational analyses confirm the roles of specific residues, offering insights for further engineering to improve antibody-specific glycosynthesis. Other papers focus on enhanced functionality of glycoengineering. A recent study introduces alpha PD-1 IRGD2, a glycoengineered PD-1 antibody IRGD peptide conjugate, as a novel immunotherapy for cancer. By combining enhanced tumor penetration with dual targeting of tumor cells and PD-1 plus T cells, this activates and amplifies tumor-specific T cells while maintaining biosafety. In mouse models, it effectively reduced tumor growth, remodeled the tumor microenvironment, and expanded a subset of CD8 plus tumor-infiltrating lymphocytes, expressing stem and memory-associated genes like TCF7 and IL-7R. Unlike traditional bispecific T-cell engagers and scFv-based therapeutics, this offers improved solubility, stability, and circulation half-life, addressing the challenges of poor drug uptake and immune infiltration in solid tumors. This platform exemplifies a promising therapeutic innovation for cancer immunotherapy, combining antibody functionality with targeted peptide delivery to overcome limitations of current approaches. Meanwhile, another study developed glycoengineered rice cell lines, using CRISPR-Cas9 to eliminate plant-specific N-glycans and produce trastuzumab with enhanced efficacy. The resulting p-Tmab showed improved anti-cancer effects, including stronger inhibition of BT474 cancer cell proliferation and greater antibody-dependent cellular cytotoxicity, ADCC, than Tmab. p-Tmab also demonstrated better tumor uptake and less liver accumulation in xenograft assays. On another focus, this study presents a novel strategy for the direct glycosylation analysis of intact monoclonal antibodies. By combining two mass spectrometry techniques, direct infusion ESI-MS for intact glycoform analysis and MALDI in-source decay for direct glycan analysis. This combined approach provides profile glycosylation in monoclonal antibodies without the slightly more time-consuming enzymatic treatments or separation techniques. The MALDI ISD-MS method can give insights into glycosylation microheterogeneity, detecting glycans such as MAN5 and G0 that were otherwise missed in intact protein analysis. It provides complementary information to traditional methods, enabling the rapid comparison of glycoforms and enhancing the analysis of glycoengineered monoclonal antibodies. The antibody industry is increasingly focused on glycoengineering as a strategy to improve therapeutic efficacy and specificity. Recent collaborations, such as the one between OB Pharma and Glytech Inc., highlight the growing emphasis on glycan production for antibody drug conjugates. As glycosylation plays a significant role in enhancing ADCC and optimizing pharmacokinetics, companies are investing in advanced technologies to control glycosylation patterns. The integration of high-throughput glycosylation profiling and gene editing is expected to further accelerate the development of next-generation antibodies, offering more effective treatments for oncology and other therapeutic areas. The industry's push towards glycoengineering, supported by strategic collaborations and technological advancements, underscores the importance of glycan optimization in the future of antibody therapeutics. Moving on to our next topic, anti-malarial antibodies. A team of researchers from the National Institutes of Health have discovered a novel class of anti-malarial antibodies that could lead to the next generation of interventions against malaria. Published in Science magazine, this study highlights a potent monoclonal antibody, MAD21-101, which binds to a previously untargeted region of the P. falciparum sporozoite surface protein, PFCSP. Unlike existing vaccines and monoclonal antibodies, which target the central repeat region, MAD21-101 binds to a conserved epitope called P-glu-CSP, exposed only after a specific developmental step in the sporozoite. This epitope, widely accessible on the sporozoite surface, is absent from current vaccines, meaning it could be used in combination with existing interventions without diminishing their efficacy. While further studies are needed to assess the activity and effectiveness of MAD21-101 and related antibodies, This discovery represents a crucial step towards reducing the global burden of malaria. Meanwhile, a study co-led by scientists from UT Health San Antonio have uncovered a critical weakness in P. falciparum, confirming that individuals in malaria-endemic regions can develop broadly neutralizing antibodies against the parasite. These antibodies target a specific site on the polymorphic erythrocyte membrane protein 1, which the parasite relies on to bind to the endothelial protein C receptor of human host cells, an interaction essential for severe malaria to develop. This site, described as the parasite's Achilles heel, cannot mutate without compromising its function, making it an ideal target for vaccine development. The findings pave the way for the development of vaccines that mimic the naturally acquired immunity observed in malarial-exposed populations. Further research aims to refine monoclonal antibodies targeting PFEMP1 and optimize computational protein design for future vaccine frameworks. Our final topic of today are antibodies and PROTACs. Proteolysis targeting chimeras, PROTACs are heterobifunctional molecules designed to degrade specific proteins by recruiting an E3 ubiquitin ligase to the target protein, leading to its ubiquitination and subsequent degradation by the proteasome. This novel mechanism enables PROTACs to eliminate oncogenic proteins, offering a unique advantage over conventional inhibitors. Meanwhile, antibody-drug conjugates have transformed cancer therapy by selectively targeting tumor-associated antigens. ADCs enhance the therapeutic index by coupling cytotoxic payloads to antibodies, allowing specific delivery to cancer cells. Thus, combining these two modalities could unlock new therapeutic opportunities. ADCs rely on internalization to deliver cytotoxic payloads effectively into target cells, but not all ADCs are efficiently internalized upon binding to their cell surface targets. A recent study by researchers from Mayo Clinic explores the hypothesis that PROTACs can enhance ADC activity by promoting their internalization through targeted protein degradation. The findings reveal that PROTACs significantly improve the internalization of antibodies and their derivative ADCs targeting oncogenic cell surface proteins such as EGFR, HER2, and MET, achieving a 1.4 to 1.9-fold increase in internalization across models. Moreover, PROTACs enhance the cytotoxicity of HER2 targeting ADCs, with these effects being dependent on dynamin and proteolysis. A recent review by Wang et al. Describes how antibody PROTAC conjugates address limitations of traditional PROTACs, such as off-tumor toxicity, poor stability, and inadequate pharmacokinetics. Notable breakthroughs include NGP41, a BRD antibody PROTAC conjugate with extended stability and robust tumor regression in xenograft models, and NGP42, which selectively degrades BRD4 in HER2+ tumor cells while sparing normal cells. Innovations also target specific antigens like STEAP1 in prostate cancer and EGFR in non-small cell lung cancer, achieving tumor-specific protein degradation and improved anti-proliferative effects. Structural modifications, such as optimized linkers and enhanced drug-to-antibody ratios, further elevate the efficacy of compounds like NGP45 and NGP46. Additionally, novel designs like NGP4851 demonstrate antigen-dependent protein degradation with cell-selective delivery, paving the way for personalized therapies targeting distinct molecular markers. These advancements underscore the promise of antibody PROTAC conjugates as precise and potent therapeutic agents in oncology. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow Biointron on LinkedIn or at our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.